Hello students, welcome to Learner's Planet. In this session we are going to discuss about the basic things that can help you in scoring better marks in your exams. We all know that there are the different patterns of the questions in your exams, isn't it? And in this subject that is biology, one of the important pattern is the labeling of the cell organelles, or labeling in any particular diagram. So this labeling is basically providing us a better way so as to score good marks, and therefore we are going to give you some tips which can help you in labeling the diagrams correctly, as well as with the help of diagram how we can just remember the functions. So in today's session we are going to give you one example of such pattern, that is a labeling of diagram, and here we have chosen the diagram of cell organelles, that is nucleus. See, in your exams, in a particular question, there might be some organelles written over there, and the examiner may ask you the function of that particular part of the organelle. Right. Alternatively, they can give you the picture as we have shown here in this particular slide, and the examiner may want you to label that particular organelle. Right. So here we are going to do it in a manner that can help you in just remembering the functions. Just recall it, and of course the part which is labeled here, we can mark it properly. So the very first structure shown here, this outer outline. Right, you can see this pink boundary over here, and this particular part or this outer boundary is of course a membrane. Any particular diagram is there, of course, especially we are discussing about the cell and its organelle. So over there, whenever there is an outer boundary, right, so of course it is going to be a cell membrane. Now with respect to a particular organelle, the name might be different. So here in the case of this nucleus, this particular membrane is known as nuclear membrane. So you can simply label this particular thing here, that is nuclear membrane. Now this nuclear membrane, in the case of nucleus, it is a double membrane structure. There is an outer membrane and there is an inner membrane. So in case if this picture is shown with two outlines or two arrows over there, let us say for example we are showing it here in the middle point, right? Now if this arrow is showing the outer boundary and the second arrow is showing the inner boundary, then there is no need to get confused. You have to simply write down as the outer nuclear membrane, or it is the inner nuclear membrane. Now this is very simple, that if it is on the outer side, that is going to be the outer nuclear membrane, and if it is on the inner side, then it is going to be a inner nuclear membrane. Right. Now we have studied this thing, that this nuclear membrane is not continuous, it is discontinuous. Now they are having some pores in between. So over this boundary you can see these particular round structures, and these round structures are known as the nucleopore. So basically what you need to do is either you just remember those basic properties and you are able to label it, or you simply just remember this picture, and with this picture we can find out the specific region, and if region is known, purpose is also clear. So this nuclear membrane, it is separating the inner and outer part of the nucleus, right? That means the matrix or the material which is filled inside, the substance which is present inside the nucleus, is separated from the cytoplasm by this particular boundary, that is nuclear membrane. But of course, since it is a living structure, right, and it has to work in coordination with other parts of cell, so it has to communicate. That means it cannot be totally impermeable. There should be some porosity, there should be some permeability, and that is provided additionally here with the help of these nucleopores. These nucleopores, they help in the communication of various molecules, transfer of various molecules in and out of the nucleus. Beside that, the important part of the cell is actually present inside the inside this particular nucleus. And what is that specific part? That is the chromatin. So this chromatin, these structures, the blue thread like structures, you can see here, these mass here, that is actually the chromatin. So we can label this chromatin here, as well as these cross-shaped structures are also shown. So these cross-shaped structures are actually known as the chromosomes. Now at this point, just keep one important thing in your mind. Here we are showing this chromatin as well as chromosomes together in the same diagram, but actually these two are not present together, right? If it is in the dividing stage, if the cell is going under the process of cell division, then definitely these chromatins, they will form these condensed cross-shaped structures which we call as the chromosomes, right? Besides, if it is in the non-dividing stage, then it is present as this entangled mass here, entangled mass of thread which we call as the chromatin. Right? Now of course, whether it is chromatin or it is chromosomes, they are having the genes which are located over it, and these genes are made up of DNA. Right? So these DNA or the genes, they are the coding fragments of the chromosomes, which is going to it is going to code for a particular protein, right? And of course, when protein is available, different functions of the cell or the body is being performed, right? Now beside this, the central region is shown here, and this central region within the nucleus is known as nucleolus, right? So this nucleolus is the another part which is generally asked to label in the diagram of the nucleus in your exams, right? Now this nucleolus is having a specific function, and what is that function? It is actually it is a site where the ribosomes are synthesized, where the RNA is present. So it is rich in RNA and ribosomes. Further, these RNA and ribosomes, they are actually transferred out of the nucleus, and they help in the synthesis of the proteins. Right. Now beside all these parts, one more important thing is present here. All these are not present just in the airy area, isn't it? The interspace is actually filled with the matrix-like substance or the jelly-like substance, which is known as the nucleoplasm. Right? And that is, of course, it must be, if it is asked, it must be present over there as a dotted structure or as a jelly substance. So if such structure is being asked to label, then what you have to write there is nucleoplasm. So this nucleoplasm is of course a very important part here. What it is doing here? It is helping in maintaining the integrity of all these parts within the nucleus. Not only this much, it is made up of water, it is having various biomolecules like the amino acids, the proteins, various vitamins, of course the nucleotides, and so on. And therefore it is providing the basic material for the existence of these particular material within the picture or within the nucleus. So this is one important thing to understand here is that this is a eukaryotic nucleus, right? So other questions which comes regarding the nucleus is the comparison of the prokaryotic nucleus with the eukaryotic nucleus. So over there in the prokaryotic organisms or a prokaryotic cell, the structure is not something like this. How is it present over there? It is, in that case, this membrane is not present, but of course the nuclear material, that is the chromosomes or the chromatin, is present over there in a particular region, and that region is known as the nucleoid. So whenever questions are coming, just keep in your mind that whether it is a prokaryotic cell or it is a eukaryotic cell, right? Now many a times they are not showing you this picture. They are simply asking you to draw the diagram of a eukaryotic nucleus, right? So in that case, you have to be clear that how this nucleus should appear. It is going to appear over there. So that diagram, drawing this diagram is even not very difficult. You can easily do that if you are going to do practice just for 2 or 3 times. Of course, you have to label the diagrams, you have to learn the basic things, and that is the basic way how you can score very good marks in your exams. Diagrams, labeling, these function type of questions, they are really very scoring. So I can help you if you in drawing this diagram also. So when you are drawing a diagram of a nucleus, you must begin with the outer wall or the boundary, right? So what you have to do, you know that it is actually discontinuous. So what you have to do is just draw this boundary. Let us just make it out, and you can draw with me also. Just pause your video and then you can continue. So this is how we have to make a double boundary structure. I'm sorry. Just make it properly so that there is no mixing. Here it is quite difficult. Let us do it. So this is how you can draw it here. Once this particular boundary is over, just keep one thing in mind. These boundaries, they should be uniform. Right. Now after that, you can draw the chromosomes inside. Just remember one thing, if you are drawing the chromosomes, then just do it. Just make cross-like structures. And if you are making chromatin, then you have to make an entangled mass of thread, right? So let us do like this. We are making an entangled mass. That means this should be labeled as what? It should be labeled as chromatin, right? Then beside this, you need to just make a region here which we are going to call as the nucleolus. Fine, like this. And beside that, you have to make some dotted scattered mass here, which is of course what? It is the nucleoplasm. So this is how we can just make it randomly. Now that now what you have to do? Do the labeling. So just label the diagram now. First part, it is the outer membrane, outer nuclear membrane. Then what? Then here is the second part, that is known as the inner nuclear membrane. Right. Then you can mark here this thing, that is the chromatin. That is the third part. Right. Now after that, you can label this particular region that we are calling as the nucleolus. I hope if there is no difficulty in this thing, because we have already marked these particular, we have labeled the diagram just now in the earlier slide. Right. Then after that, you can label this scattered mass, the jelly-like substance, which is known as the nucleoplasm. And of course, the next part that we need to label here is the nucleopore, that is the spaces in between. So this is how you can draw the diagram so easily within 2 minutes. You have to practice it in a way that you are not going to take more than 2 minutes to draw any diagram with labeling, and that is possible only with the help of the only through the practice. So this is all about the nucleus, and we can just understand about some other organelle in the next session. So thank you so much. Have a nice day. We'll meet you soon in the next session.